And the next group of biological molecules we'll talk about are lipids. And lipids are a diverse group. You can see that they include fats, oils, waxes, phospholipids, and steroids, and we'll talk a little bit about each of those. But one thing that I want to make sure I point out is, even though these are a diverse group of molecules, they are similar in that they're all hydrophobic or water-fearing. Remember, hydrophilic means water-loving, things that easily dissolve in water. Hydrophobic would be the opposite of that. So when you think about when you mix, if you make salad dressing with oil and vinegar, it doesn't matter how many times you shake it up, they're still going to separate into those two separate layers. So one thing to point out is you can see from the figure here, for example, this triglyceride right here is what we will also call a fat. You see there's a steroid structure here and a phospholipid structure here. The thing that you see in common, these are different structures, but you can see that they are made up of mostly carbon, right, and hydrogen. And so we would call that a hydrocarbon. And so what is oil? It's a hydrocarbon. Okay, and we know that oil and water don't mix, so lipids definitely have a high component of hydrocarbon. Let's start with fats or triglycerides, and they're named that. The tri means three, and it's talking about three fatty acids, and these fatty acids are bonded to one molecule of glycerol. Now, we've spent a lot of time so far talking about monomers, okay? In this case, each of the fatty acids would be a monomer, okay? And as they're bonded onto this glycerol molecule, okay, that's the building up of the polymer. So in other words, this one fat or one triglyceride would be the polymer, and the individual fatty acids in the glycerol would be the monomers. As we go through and talk about fats, you probably know from reading labels or something about diets that there can be unsaturated fats and saturated fats, and you may already have an idea that somehow unsaturated fats are better for you than saturated. We're going to understand that chemically what that means is there's a chemical reason that we call something unsaturated, and that has to do with the fact that there's a double bond between carbons in that molecule, and we'll look at specifically what that looks like. The other thing I want to say about fats is that sometimes they're referred to as energy rich. And the reason that it's depicted that way is they store a lot more energy. We measure that in calories, so a lot more energy per gram than other things. So for example, when you consume carbohydrates and proteins, for every gram you consume, that gives you about 4 calories per gram. When you consume fat, for every gram of fat, it gives you about 9 calories. So that's more than twice as much. But if for us, when we typically in the United States, right, are watching not to have too many calories because we have such a problem with, obesity in our country and the health problems that it causes. The other way to look at this too, though, is fat is a long-term energy storage molecule. So if we're thinking about storing away a certain number of calories, right, it takes less grams of fat to store those numbers of calories away than it does if we stored those away as carbohydrates or proteins. Now let's look at the chemical structures of these fatty acids that form a fat. Okay, so this up here would be one fatty acid. Okay, and this would be a separate fatty acid. So remember, it takes 3 fatty acids bonded to that glycerol, and they can be, a wide variety of different fatty acids, and we just have 2 examples for you here. The top one is a saturated fatty acid, and the reason I know it's saturated is essentially it's saturated with hydrogen atoms. Okay, so remember, besides this little portion here, all the rest of a fatty acid is just a hydrocarbon. Well, we know that carbon is always going to make 4 bonds, okay? So every carbon in this chain is making 4 bonds, okay? Now, each of these carbons, for example, except for the terminal carbon here, right? One of those bonds is made with the neighboring carbon here, and one of those bonds is made with the neighboring carbon there, which means that leaves 2 bonds left, so it's going to be bonding with 2 hydrogens. Now, if we look down below, what we see is in an unsaturated fatty acid, we have a change there. So in this one little spot, instead of the carbon being bonded to the neighboring carbon with a single bond, in this case, it's actually 2 bonds. So what that means is each carbon can still only make 4 bonds. So if we look at this particular carbon right here, okay, that's 1, 2, 3, 4 bonds that it's making. So the hydrogen that would have been here, right, it can't bond with that because it's already making 4 bonds. So this up here we say is saturated with hydrogens. In other words, it's bonding to the maximum number of hydrogens. Anytime you introduce a double bond, there are less hydrogens there as part of that hydrocarbon because of the carbon-carbon double bond. Now typically, chemically, when you have a saturated fat like this one, okay, that's going to be a solid at room temperature, like butter. And typically, if you have unsaturated fats, those are going to be liquids at room temperature, like oils. I also want to point out that there are a couple of fatty acids. So fats are not all bad, right? We certainly need fats in our diet. And as we go through the rest of this section and we talk about steroids and we talk about phospholipids, you'll see that fats play an important role in our cells in addition to being an energy storage molecule. Some of these fatty acids that are needed to make these certain types of triglycerides or other fats or lipids, we have to get in our diet. We can't synthesize those in our cells. We have to eat those. So linoleic acid and alpha linoleic acid are 2 of those, what we call essential fatty acids. We say essential because we must consume them in our diet. So here's something else I want to make sure that I highlight for you, and that is something that you're probably familiar with, something called trans fat. So we talked about saturated fats, we talked about unsaturated fats, and now we're going to hit on trans fats. So a trans fat is actually something that is man-made, okay? It's created for a purpose. And what they do to create these trans fats is they take an oil or an unsaturated fat, something that's not solid at room temperature, but they add hydrogen atoms to it. Remember, an unsaturated fat means it's not saturated with hydrogens, right? It's got some double bonds in there. So they partially call it hydrogenated, and you might see on food labels it's called partially hydrogenated, right? That may be on the label. By adding a certain amount of hydrogens to that molecule, it's a couple of things that is good for shelf life of food, right? First of all, it makes it more solid, which is good when you're making crackers and cookies, and also it gives it a longer shelf life. It doesn't break down as quickly or become rancid. But what it does do is it actually makes the chemical structure different. So if this up here is an example of a saturated fat, you can see the structure, right? There's no double bonds. So in nature, when we have unsaturated fatty acids, they have this, it's called a cis configuration, which has this natural kink in it. And the enzymes in your cells recognize that kink because that's the typical structure of an unsaturated fat. However, the man-made trans fat version are shaped differently, so they don't have that kink. And the enzymes in your cells don't recognize this structure readily like they do the other, so they don't get broken down, and they can go through your bloodstream and begin to cause issues, right? Build up, which can lead to heart disease and high blood pressure and other things like that. So trans fats, as you probably already know from things that you've read, are not good for you. Here's just some example of different types of oils. So it's showing you how many grams per 100 grams of this, per 100 grams of fat are saturated versus polyunsaturated versus monounsaturated. So mono meaning one point of unsaturation or one double bond, poly meaning multiple. So you can see, remember I mentioned to you that linoleic acid and alpha linoleic acid are actually 2 essential fatty acids. So you can see based on this chart the composition of the types of fats that are found in those particular oils or other fats. Now let's move to phospholipids. So a phospholipid, I have the structure here for you, looks very similar to a fat in that, okay, first of all, we have this glycerol molecule that we had previously with the fat. Now, in addition to that, we have a phosphate, right, which is why they're called phospholipid. Whoops, yep, phospholipid, there we go. But we have this long hydrocarbon chain, right, just like we had with a fat. The difference is there are only two of these long fatty acids or long hydrocarbon chains that are coming off of a phospholipid. Now, what's important about phospholipids are they make up the majority of plasma membranes for cells. So these are very important, right, to us and to all living things because all living things have plasma membranes. Now, chemically, what's going on is they have part of their molecule that's hydrophilic or water-loving, and part of the molecule is hydrophobic or water-fearing. So because of all of these polar atoms like oxygen and phosphorus up here in the head portion, this is going to be the hydrophilic area, or the part that's going to be able to readily dissolve in water. Down here where we have all the hydrocarbon, which is oil-like, it's going to have a hydrophobic nature. So within that one molecule, part of it's hydrophilic, part of it's hydrophobic. Now, these come together in what we call bilayers to form plasma membranes. And as you probably are seeing already, sort of like attracts like. Things that are hydrophobic want to be around other hydrophobic molecules, right? Things that are hydrophilic stay around other hydrophilic things, as in the salad dressing, right? The hydrophilic and hydrophobic repel and don't mix. So what that means is when you have multiple phospholipids together, the heads are going to come together, right? Because they're hydrophilic. The tails are going to orient together because they're hydrophobic. So this would be one layer of phospholipids. This here would be a second layer. And the second layer orients so that the hydrophobic tails orient with the hydrophobic tails of the other layer, and the hydrophilic heads orient together. Now what we know is that the inside of cells is mostly water, and the fluid outside of cells is mostly water. So if you can imagine then that if this were the plasma membrane, okay, this would be inside the cell, right, which is mostly water, and this would be outside the cell, which is also mostly water. So you have the hydrophilic heads that like water pointing in, right? You have the hydrophilic heads that like water pointing out. And then you have this sandwich in the middle of the hydrophobic tails that are coming together. So as we'll see as we move on in the course, the fact that you have these hydrophobic tails as part of the plasma membrane helps regulate what can come in and out of the plasma membrane. Now let's talk about steroids. So these steroid structures, as you're looking at them, so cholesterol, testosterone, which is we think of as the male hormone, and estradiol or estrogen, we think of as the female hormone, okay? These all have this 4-ring structure. So we have like 1, 2, 3, 4 rings with some other things, right, bonded off, coming off that change, but the 4-ring structure holds true. So when you're looking for steroids and you see, or looking at structures and you see these 4-ring structures, it's going to trigger your memory that must be a steroid. So these serve as hormones, right, signaling molecules that travel through our bloodstream. We think of typically as cholesterol being bad, right? We talk about cholesterol being too high or foods that are high in cholesterol, but actually cholesterol is important in the plasma membrane. So it causes the plasma membrane to not be too rigid or too fluid. It's also the precursor for these male and female hormones that we certainly need. And it's also the precursor of a couple of important vitamins, vitamin K and vitamin E. Now you may have heard of, or you may have had a blood test and they give you reports on your good cholesterol and your bad cholesterol. So what they're actually telling you is not simply a measure of cholesterol, but there are carriers of cholesterol in your blood, okay, called low density lipoprotein, or LDL, and high density lipoprotein, or HDL. And the reason that these are considered bad and good is just dependent on where these carriers are taking the cholesterol. So the LDL, we call it bad cholesterol because essentially it's taking it into the bloodstream, right, and circulating this cholesterol, which can lead to, right, a buildup or plaque or heart disease. HDL, or good cholesterol, is actually carrying it, is returning it from the bloodstream, so it's taking that cholesterol out of the bloodstream, so that's considered, a good form of cholesterol. Now, I want to end with a blank version of this chart that we started with at the beginning, and I'm going to provide this to you in the lesson, and it's important now that you try to go through and identify the monomers of each of these, some examples, the polymers of these, and some examples of those. And hopefully at this point we've put the package together that there are 4 main biological molecules or macromolecules. They're all carbon-based or organic. They serve an important role in the cell, and they're all based on a particular building block or a monomer. And those monomers are bonded together through dehydration synthesis reactions to form these larger molecules. And these bonds that we're talking about that form these larger molecules are very strong covalent bonds in the cell.